vitamin is your vitamin B12. And what is the vitamin B12? It is also called as a cobalamin. Okay, cobalamin. Vitamin B12 is also known as a anti-pernicious anemia. Okay, vitamin. So it's very very unique vitamin which is synthesized by only microorganism and not by the animals or not by the plant. It was the last vitamin to be discovered. Okay, this was the last vitamin. So basically, what is the chemistry? Vitamin B12 is the only vitamin with the complex structure. If you have seen, other vitamin, they do not have so much of complex structure. So this is B12 is the only vitamin with so much of complex structure. The empirical formula of vitamin B12 is also called as it is called as a cyanocobalamin. Okay, cyanocobalamin, and its empirical formula is atoms number of carbon. Okay. Okay, so it is C63, H is 90, and N is 14, and O is 14 2, and right, it is so complex. Okay, it is called as a cyanocobalamin, and the structure of vitamin B12 consists of a corine ring. You see, this is the corine ring with a central cobalt. This is the cobalt atom. Okay, this is the corine ring with the central cobalt atom. The corine ring is almost similar to, you see, tetrapyrrole ring structure which is found in other porphyrin compounds like you remember hemi, or you have not yet studied hemi or myoglobin, or you must have studied chlorophyll. Okay, so all these chlorophyll, myoglobin, all of them, they have, this corine ring is very very similar to this tetrapyrrole ring. This is the tetrapyrrole ring structure which is also found in like myoglobin, hemoglobin, and this chlorophyll. So porphyrin ring compounds, they are called as a porphyrin ring compounds because they have this ring structure. Okay, so myoglobin, hemoglobin would have Fe, chlorophyll would have this magnesium. Okay, we will see soon in the next lecture. I will be taking this. So this corine ring has a 4 pyrrole units. You see, 1, 2, 3, 4. So pyrrole units, just like a porphyrin ring that is present in the hemi and chlorophyll pigment. Two of the pyrrole units, like the out of these two of the pyrrole units are directly bound to each other, and whereas the other are held by methylene bridges. So, and the group namely methyl acetamide and pro propionamide are the substituents on the pyrrole ring. So vitamin B12 has this cobalt in a coordination state of 6, and cobalt present at the center of the corine ring is bonded to the 4 pyrrole nitrogen. 1, 2, 3, and 4. The 4 pyrrole nitrogen. Cobalt also holds below the corine plane. Okay, so that is that, you see there is also dimethyl, where is the dimethyl benzene amidazole. Okay, so DMB, which contains ribose 5 phosphate, it's very very complex. Okay, and amino isopropanol. Okay, so it is connected here also. This cobalt also holds this DMB, that is dimethyl benzene imidazole compound, that is DMA, and which contains this ribose 5 phosphate and amino isopropanol. Amino isopropanol. 1, 2, 3, 4 ribose. Okay, amino isopropanol. A nitrogen atom of dimethyl benzene imidazole is linked to cobalt. The amide group of amino isopropanol binds with the this ring. Okay, we generally it is named as like this A then this B, and you see there are this C and then this D. A, B, C, D. This is the ring. Okay, this 1, 2, 3, 4. Okay, so this is also connected here. The group of this amino isopropanol binds with this ring also. You see here it is also connected here. Okay, so the cobalt atom also possesses a sixth substituent group which is located above the plane of Corrin ring. Okay, so it's so complicated. The substituent group may be one of the following, like it can be either cyanide that is predominant in cyanocobalamin. Okay, uh, then hydroxyl in hydroxycobalamin, or it can be nitrite in nitrocobalamin. So there are two coenzyme forms of vitamin B12 observed, and the deoxydeonacyl that is cyanide is replaced here, and methyl cobalamin. Okay, so cyanocobalamin, which is a synthetic form of vitamin B12, generally used in food system for fortification and nutrient supplementation, and it exhibits superior stability and it is more readily available commercially. Coenzyme forms of vitamin B12 like methylcobalamin and 5-deoxyadenosylcobalamin is also there. So this methylcobalamin, they are the coenzyme form, okay, that function as coenzymatically in the transfer of methyl group in the methionine synthetase reaction, while this 5-deoxyadenosylcobalamin also acts as a coenzyme in an enzymatic rearrangement reaction which is catalyzed by methyl alanine coenzyme A mutase. Okay, so basically this cyanocobalamin has a reddish color and it is present in the crystalline state in the solution, and this is the limitation where it is not possible to add cyanocobalamin into certain food because then it gives that reddish color to the food. For example, if you want to add the cyanocobalamin in white bread, it's not possible, then the bread will become reddish in color. So there are food according to their B12 concentration. For example, vitamin B12 more than 10 milligram per 100 gram. So they are called as a rich sources of vitamin B12. For example, non-vegetarian sources such as liver, kidney, heart, clams and oysters are very good source of vitamin B12, and that is why vitamin B12 deficiency is observed into strict vegetarian people. Okay, because this is in high concentration in the non-vegetarian sources. There are moderately resources such as non-fat dry milk, some fishes, crabs and egg yolk. So the vitamin B12 ranges between 3 to 10 milligram, okay, per 100 gram of these moderately rich sources. There are also moderate sources for whom the vitamin B12 ranges between just 1 to 3 milligram. For example, some fishes, fermented cheeses, and less than 1 milligram, like very poor sources like fluid milk, cheddar cheese or cottage cheese is not a very good source of vitamin B12. So under food processing conditions, preservation, storage, transportation, there's little nutritionally significant loss of vitamin B12 observed. Cyanocobalamin added to the breakfast cereal product has been reported to undergo an average loss of around 17% during processing, with an additional 17% loss during storage, okay, and ambient temperature. Generally breakfast cereals we keep it at ambient storage, so almost 35% of the loss is observed, total 35%, 17% during processing and remaining 17% during storage. In studies of processing of fluid milk, 96% mean retention was observed when high temperature short time treatment or pasteurization was done. So pasteurization does not, deactivate or degrades the cyanocobalamin. Greater than 90% was found in milk processed using ultra high temperature, where the temperature is high, but the period is like quick, like few seconds. Further, refrigerated storage of milk has little impact on vitamin B12 retention. So, a study was carried out wherein the storage of this UHT processed milk at ambient temperature for 90 days was performed, and it was observed that a progressive loss was caused, and that can approach 50% of the initial. Basically, milk is a very, very poor source of cyanocobalamin. So, photochemical degradation also observes with respect to vitamin B12 coenzyme that results into aquacobalamin. The overall stability of vitamin B12 is greatest at pH 4 to 7. We know that vitamins are quite sensitive to pH, and in this case, pH 4 to 7 gives rise to a very, very good stability. Exposure to acid causes hydrolytic removal of the nucleotide moiety, and additional fragmentation occurs because of the severity of the acidic conditions. Exposure to acid or alkaline condition causes hydrolysis of amides, yielding biologically inactive carboxylic acid derivatives from the vitamin B12. So there was a study in which vitamin B12 bioavailability was examined, mainly in the context of diagnosis of vitamin B12 deficiency, which was associated with the malabsorption. And several studies have shown that the pectin or similar gums reduces the B12 bioavailability. So basically, pectin, fibers, gums are complex polysaccharides which binds with the vitamin and make them unavailable. So because they are complex carbohydrate or they are fiber, and therefore the bioavailability is reduced. The significance of this effect in human is unclear. However, little vitamin B12 is also present in most of the plants, like in the form of algae, and algae is not recommended as a source of vitamin B12 because of bioavailability, because of the there are other compounds that makes binds and makes it unavailable. Okay. However, in normal human beings, absorption of B12 from eggs has shown to be less than half that of the cyanocobalamin administered in the absence of food. And there are a lot of studies have been carried out wherein, poor digestion has caused vitamin B12 deficiency, or incomplete release of cobalamin from the food matrix can also result into deficiency. So again, similar to other food system, other vitamins, there are analytical methods based on microbiological assay, or HPLC methods are also could be explored. But again, food samples should be homogenized and buffered in a proper solution. They should not be used or exposed to high temperature, which will result into